All right, I think we can start. Welcome everyone to our physics colloquium. It is my honor and pleasure to introduce our esteemed speaker today and guest from the School of Engineering, Professor Kimani Toussaint, Jr., who is currently also the Associate Dean of, I don't know if I get it right, Science and Research Strategy. Yes. So very important.
Something like that, that's right.
Very important Associate Dean, yes. And so to introduce Kimani Toussaint, I will say, so he did his undergrad at the University of Pennsylvania. He was a roommate of someone in the audience that I leave to you to find out because I'm not supposed to say this. Then he moved on to get his PhD from Boston University in electrical engineering, after which he did a postdoc at the University of Chicago working on super resolved optical imaging and optical tweezing, then became faculty at UIUC. First assistant professor up to full professor, and then we were lucky to have him come to Brown in 2019, and he runs a very productive research group here, and we'll hear about some of his work today that uses femtosecond optical tweezing specifically, and then plasmonics, where he made a lot of contributions. To mention a few of his awards and honors, he's a fellow of SPIE, which is for those who don't know, Optical Engineering Society of Optical Engineering. He's also a fellow of Optica, former Optical Society of America, and a few other. He has a career award, of course, and number of patents in this field. So a very esteemed speaker today. And without further ado, I'll give the floor to yours, and I look forward to your talk.
Thank you. So I was telling my colleagues in physics that I kind of like this tradition, right? So I teach in this classroom, and normally when I give seminars, I travel, not from the second floor to the seventh floor or the third floor, but travel, get on a plane, go somewhere else. So it's kind of nice to treat this formally and get to know some of the colleagues in physics, although of course engineering colleagues are here as well. So that was very nice. So I thank physics again for inviting me. What you'll see with this talk is sort of the potpourri of ideas that goes on in my head and my students and their patients in exploring some of these ideas. We have quite a few interests, and over the years these interests have evolved somewhat. I will highlight just a couple of things here, and I'll give you a snapshot of some aspects of what we've done over the years, with a particular focus eventually on the two-photon polymerization stuff. But I actually started off. I guess my connection to physics is that I started off for my PhD working in polarization entanglement, using parametric down conversion, and that's all tabletop experiments meant to explore the fundamentals of fundamental concepts in quantum physics. I was in engineering, so we were looking to make applications out of it. Outside, of course, quantum computing, I focused on polarization entanglement. When I started off as a faculty member, and fast forward to the postdoc and so forth, I was curious about sort of that reverse process. So rather than having a single high-energy photon enter a nonlinear medium and generate pairs of entangled photons, we have these low-energy photons get together and create this higher energy photon in the form of second harmonic. And that'll open up the door for us to do bioimaging, which is something that I've been interested in doing. Then at the same time, when you have a multi-photon platform, microscope in our case, that allows you to do other types of metrology. So I had a colleague who was very much interested, especially when plasmonics was huge, in looking at the nonlinear properties of plasmonic materials, and I focused on, and that kind of introduced me to plasmonic antennas. So I'll say just a couple of words about that. Because we're optics people, we like to do stuff with light, like program the light to have orbital angular momentum, or for it to be something called a vector beam. So we explore some of this as well. Recently, we've been looking into using our multi-photon platform not just for reading information, but for writing information in the form of two-photon polymerization. There's been some nice advances there, looking at potentially enabling the ability to do in situ mechanical measurements and testing. I can't talk about everything today, unfortunately. I will bore you all. But I will talk about some aspects of some of these things. And something that's grown quite considerably in the last few years, in large part because of the pandemic, is no fancy lasers, but work with biosensing, trying to make an optical sensor that can read your blood oxygen content in a way that's independent of your skin tone. And this is what's caught on in terms of the broader public. I won't talk about that aspect of it today, but I will try and talk about the other things. And the fortunate thing is that I'm here, so if you all want to ask questions, you can always find me. All right, so I'll talk about our work in quantitative second harmonic generation. This is what I started off when I was an assistant professor. Since I was talking to physics, I decided to throw in a little bit of the work we were doing on the Muri project on so-called non-separable fields. The controversial title for that is classical entanglement, and I'll explain what I mean when we talk about that. And then I will talk about what we're doing with this new platform of combining tweezing with two-photon polymerization. So as a bit of motivation, when I got into bioimaging, I was interested in looking at systems where I can get good signal. Right? I'm not a biologist. I wanted something where the response will be significantly high enough. Turns out that collagen is important to biology, and I have plenty of colleagues who can tell you why. But it forms the structural backbone of many things with mammalian systems. And what happens is that there's a potpourri of types of collagens, and one can think of them as having different levels of crystallinity. And in terms of collagen molecules getting together with other collagen molecules and forming some type of hierarchical structure that might be in your eye, the sclera part of your eye, or it might be in your cornea, or it might be in your skin in terms of how they organize, how they get together, correlating structure and function is an important long question that is asked. And so we decided to venture into this idea of quantitative imaging, being able to leverage the imaging technique in a way that is quote unquote label free, and that allows us to marry some type of rather simple, I think, quantitative markers that can be used as biomarkers associated with it. So when we were in Illinois, we looked to this idea of second harmonic generation. I'm sure many of you are familiar with it, so I'll go through a very, very high level. This is a simplified Jablonski type diagram where you just have electronic transitions that are mediated by these virtual levels, right? So you have you can think of it as this two-level system, but you have these intermediate virtual states. So you have light comes in of a certain frequency, let's say red photon. It excites this system to a higher virtual state. And because this is virtual, this is a superposition with the field that comes in. And then it gets excited again to this next virtual level. And then it immediately relaxes. That's on the order of, we're talking about lifetimes on the order of femtosecond scales. That's different from multi-photon fluorescence, for example, where you'll have the excitation to this real transition. And you see these ladders there, so there's other stuff that, in other words, they're non-radiative pathways, in which case you lose energy. The photon that process is no longer quantum mechanically coherent, if you will. And so the emission now is isotropic. So we look, this is P here is the polarization density. Chi is the susceptibility. So second harmonic generation relates to the second order field if it's intense enough. And you can send in now, this is all scattering, no absorption. Send in red photons and get out a blue photon, for example, right? Why would you want to do that, right? Because one of the goals with imaging is, resolution, contrast. You want to be able to send in light at a particular frequency, bring in a spectral filter of a different color, reject the probe light, and then read out what you want. So that was part one. Part two was this simple idea that, and this is a picture, of course, of Fourier, of just using spatial frequency analysis to try to get some estimate of the collagen fiber organization within a tissue. Very simple idea, which, interestingly enough, this was back in 2000, and people who do X-ray crystallography and so forth do this all the time. But this was not common in multiphoton imaging, yellow second harmonic generation imaging, back in 2009, 2010. And we did this, and I'll say one more thing. One photon versus two photon here. In case you're curious, twofold, so infrared light penetrates more deeply into tissue, right? You have less absorption, you have more scattering. So me shining a green laser pointer on my finger, you can't see anything. If I shine a white light here, you'll see my finger light up red, right? So that's a great thing. And what we like to do is not kill the sample in principle or have this huge wake of electromagnetic energy there. You can't tell from where you are, but you have basically a little green dot there. Trust me, this is a very famous picture taken from Cornell on the two photon effect versus the traditional one photon excitation process. So we said, okay, we'll combine. We know that it turns out collagen, type 1, crystalline enough that it can naturally give rise to a second harmonic signal. And we wanted to combine that with spatial frequency analysis. We wrote paper after paper over the years on some tricks you can do there to be able to determine information about how the collagen is organized within a tissue. So you can maybe discern between healthy tissue, and in this case, this is so-called equine athletes. So that's a fancy way of saying racehorses, right? So where basically you have some engineered tendon injury, where you see here, these are the collagen fibers for this tendon tissue. They show up naturally without any fluorescent markers. Here is disrupted in such a way that it mimics tendon injury. You see that naturally just from using the second harmonic. My rule of thumb, unless you're really a true image processing person, and I don't claim to be, if you see differences in the images, it's fairly easy to pick that out quantitatively. And this is what we showed. And we show that you can extend this, of course, naturally to 3 dimensions, because what multiphoton buys you is the ability to write in 3 dimensions, with the trade-off of it being, for example, slower, with some caveats, okay, with some other caveats there. And so as a result, what we did over the years was we said, okay, great, we can quantify things like collagen orientation, density, and so forth within a structure where you may have some tendon sample cut at different angles, and we can say something about certain moments that are observables of interest to us, such as how highly organized. We'll say something about the spatial entropy. It's the fibers are all aligned in a particular direction. We'll call that isotropic versus it being organized in a variety of different directions. And we apply this to a variety of different problems, something like preterm birth, as an example, where the cervical microenvironment is very rich in collagen. People don't know why some women give birth very early, a very early gestational time point, compared to others. It's still an ongoing issue. And so this was a situation where collaborated with researchers who are biomechanics folks who were doing instrument and nano indentation, someone else who was doing quantitative ultrasound, backscattered ultrasound, to look at the cervical microenvironments, in this case of rats. You see the circumferential fibers, you see this region, this bridge along this is something that's called the septum between the circumferential fibers, and you have this apparent dark region. It's not really dark, but when you look at it, you think for once that it's actually dark. And being able to now combine that with nano indentation, so you basically have a probe that traditionally was used for materials, for imprinting on hard materials, and adapted to look at soft tissues, and you can get things like Young's modulus or some estimate of that, and we can start to correlate some mechanical information with some structural information using some of the metrics that I said earlier before. So we can calculate information pertaining to orientations of fibers within the plane versus outside of the plane, and we can correlate that to the mechanical information of interest. Now, as time went on, we said, and this was something that started right before the pandemic, actually, when I came here. And we met researchers who do tissue engineering. They stumped us with much more complex tissue structures. And we were like, oh, okay, our traditional approach doesn't work as well. And I said, well, can we, when we do the math and we're looking at these fibrous structures, at the end of the day, what we have are sort of what I call pseudo vector fields, right? So you basically are calculating the local orientation. You can think of it as almost like a gradient analysis, the local orientation of the fibers, preferred orientation of the fibers within some pixel or ultimately within some voxel. So in the end of the day, if you get a field, and if you want to pretend as if it's a pseudo vector field, we said, okay, well, can we treat this as the way you would treat other vector fields, whether it's Maxwell's equations or Navier-Stokes equations, and borrow some of those observable measures from there. Turns out people already do that for different fields, right? There's a whole bunch of work that's been done for quite some time doing that. So we said, okay, great. Maybe we'll borrow some of that analogy. And sometime during that pandemic, we were able to just show that we can get, we can think of these images of these fibers and these fiber-like structures as being a type of fluid. Now, I would not say that in front of my fluids colleagues, right? But so there's no velocity here. These are static images, right? So where we can calculate things like tortuosity, how wavy a fiber might be, or we might calculate something that has to do with a type of vorticity, right? So do the fibers kind of curl around themselves? Or we look at local fast changes in the fiber orientation, and we collapse them into these particular metrics of interest. And you can do things like that. It's fascinating. This image right here, those are, that's bone, cortical bone. And these cortical bone has something called Harvesian canals, where you have these lamellar sheets of fibers that literally wrap around, right? So the metric that would correspond to that curl operation pops right up, and it allows us to distinguish as a group, as a collective organization, the fiber behavior within the tissue. Yes?
The imaging, is it seeing the bulk of a collagen, if you will, or is it seeing kind of the surfaces of the fibers?
No, it's a great question, great question. So the way you want to think about it, maybe I'll do this one here. So I skipped over a few things, right? So in optics, so fundamentally, we are always limited. There's always this competition between absorption and scattering, right? So there's a question of penetration depth. How deep can you go, right? And a lot of times, and again, I'm skipping over a few things, you work in what's called the therapeutic window. So that's roughly somewhere between 670 nanometers and around 1,000, 1,100 nanometers. That gets you within a certain depth with reasonable resolution. So 2-photon fluorescence microscopy, that's the advantage. You get resolution at depth. It's not higher resolution than what you would get if you go to shorter wavelengths, because there's something called the diffraction limit. So what limits this are scattering events, how you prepare the sample, whether or not we really put a lot of wax or paraffin wax. We like to look at our tissues sort of fresh, if you will, under the microscope. So a lot of these images would be maybe a few microns right under the surface, and you proceed further down. And so the typical resolution you're thinking about in terms of spatial, you're looking at roughly, if we're coming in with, let's say, 800 nanometer wavelength in the near IR, you're looking at roughly pushing about 400 nanometers in the transverse dimensions and on the order of the wavelength along the axial direction. And that's different from super resolution techniques and all those other things. Thank you. No, good question. All right. So what else can we do? Well, I said that this process is coherent. And what I meant by that is that the photon that's generated from second harmonic remembers how it was born, right? Because none of the energy is lost to the lattice, if you will. So what happens That means we can exploit polarization effects and try to see whether or not we can also obtain information. So forever, the people in the semiconductor industry do this all the time with things like ellipsometry. And of course, it's been adapted, right? Where you may have a system of interest. And what you do with that system of interest is you perturb it with different states of input polarization. And then you look at the projections along a different set of polarization directions, right? So that's pretty much cookbook. And if you're careful with it, you can build up vis-a-vis a phenomenological model. That's important to also understand something called a Mueller matrix that maps what we refer to as a Stokes vector at the input to a Stokes vector at the output, right? But you can get all the information you want out of it. In ellipsometry, people normally do this to get like thickness of some layer or multiple layers with some initial assumptions about what's in there from that system. And they would simplify. You don't need Mueller matrix if you don't care about depolarization or anything of that sort. So we were curious about sort of adapting that technique because there are standard observables then that we can maybe use to try to quantify our structures and things like depolarization, dye attenuation, retardation, whether it's linear or circular retardation, and so forth. There's a whole bunch. And so what we found was that when we do this, we can basically incorporate polarization state generators and analyzers into the system. These are general, so you can generate any state of polarization that you want. Put that onto your system, and you're going to get some interesting measurements. But the problem was that, one, I'm again skipping over a few things. We had to generalize our Mueller matrix to a 2-photon Mueller matrix as opposed to a traditional 1-photon Mueller matrix. And concepts pertaining to retardation and so forth, because you have a frequency conversion, were not as easily understandable. So what we did was something else. We said, okay, well, can we kind of build 2 or 3 systems on top of the same microscope, right? So can we use the second harmonic, which is sensitive to non-central symmetric structures like collagen, to identify them? Can we use the pump wavelength itself to combined with confocal for which to do polarimetry? And we combine them into a common platform. And from there, and again, this is all label free, we were able to identify the collagen-rich regions as well as with simple subtraction, really, as well as the images that are not collagen-rich. You can get both. Importantly, because this is all done with the pump, then you can do the polarimetry at the same wavelength, right? And so that allows us now to get the traditional observables that they use in polarimetry and apply this to interesting samples. I say interesting samples because this is trivial for biologists. For us, it was a big deal. We can purchase a nice slide that has been graded by a pathologist whom we collaborated with. This is healthy, for example, and this is not healthy. And we can compare that to the standard H&E stained images, which is what bio folks will look at under the microscope, and then determine from there the polarization information. Okay, so that was fantastic. We were happy with that. You know, it was great. And then I had students, like one of my former students now, Zishi, who's in the audience here, who pointed out that there's more to it because depending on how you do the math, how you do the Mueller matrix decomposition, you can get different results, right? So there are things to consider, you know, that there are assumptions that are being made about cumulative effects. If you have some tissue specimen that has some thickness and you propagate the field there, you're in one model, let's say this polar decomposition model, we're assuming that, for example, depolarization might happen first, and then retardation might happen next, and another effect might happen afterwards. These things don't commute. It matters. You know, you're going to get a heat map. You're going to get information. But again, interpreting that is a challenge. Or you can do something else where you try to take the cumulative sum, you know, as you go through a stack in 3D of the polarization information now to try to determine what that little slither of materials polarization response might be. So this is something that right now is ongoing. There's challenges in decoding it, but because of enough of you in this room who do biophysics related work, I want to at least let you know that we're here and we can collaborate and we're interested sort of in exploring these ideas further. All right, so I'll just put that up briefly. So that's one snippet. That is this idea of doing quantitative multiphoton imaging all around second harmonic. We extended this to third harmonic generation. It works. It's not that difficult as well. There are tricks you have to worry about with the detector and the wavelengths, but otherwise it works fine. Now, here's another area that's a little bit even more esoteric, a lot more esoteric actually, but I want to say a few words about it. This field of non-separable optical fields, which is also has been referred to as classical entanglement. Okay, and I'll explain what I mean. So there's been tons of papers on classical entanglement. They go back to at least when I was a graduate student. Even I was oblivious to these papers, and I'll tell you why this is interesting. This is interesting because there was a realization that the mathematical machinery behind quantum mechanics, putting in some type of Hilbert space, for example, can be exploited to understand some properties associated with classical fields, especially partially coherent fields. So here in the traditional model, of quantum entanglement, you have some system, for example, that might contain two particles. So that system might be represented by some state vector. And we have sort of this so-called bipartite model where you cannot describe the state of your system as a simple tensor product between the two states, A and B, okay? And we have to write it then in terms of some type of superposition state. And importantly, in the traditional, this was the stuff that I worked on when I was a graduate student, importantly here, quantum entanglement is all about non-locality, right? That's the important thing here, right? There's this spooky action in the distance thing. It's all about non-local interactions, okay? Now, it was realized that the superposition thing, adding things in such a way that they can't be just described as just products of one another in terms of their state vectors, can show up in optics. And it does show up in optics. It's just that we just don't always think about it. And so there were some really smart people who had some very creative approaches to this. And he said, well, there's no longer a bipartite system or multipartite, depending on what your flavor of entanglement is. But rather what we talk about now is the degrees of freedom of the optical system. Right? So a degree of freedom, for example, is the polarization degree of freedom, or the spatial degree of freedom, or the momentum or temporal frequency degree of freedom. And you can have states such that you cannot write them in terms of their product, but rather you have to write them in terms of a superposition. And that's been done. It's been done before, except now it's being revisited through these eyes. Right? And so this is subject of a Muri grant that I'm on. So I'll say something about some implications of that and what we're working on in the lab. So this was something that I got exposed to when I was a postdoc, this idea of vector beams, right? And even though I know, at least in engineering, and I'm pretty sure in physics as well, when students learn optics and they learn Maxwell's equations, you work with a traditional TEM wave, right? Well, it turns out, and we think of polarization as being uniform across the wavefront, right? But you can have polarization states that are non-uniform across the wavefront. It's just that what they do is they create singularities, right? So what happens in one of the most famous cases are these so-called vector beams or vector fields, right? In which case, I'm sure many of you have seen this, and this is just written in this simple case as a superposition of two Hermite Gaussian modes, right? So something like a butterfly like this plus a butterfly like that. They're coherently added, right? So there's a definite phase relationship between them. They generate a singularity because the polarization state on axis is unknown. What's interesting about something like the radial polarization or the radial vector beam is that if you focus that with very strong lens, so-called high NA focusing, you get an enhanced longitudinal field, a Z polarization. And in microscopy, especially super resolution microscopy, which was what I was working on when I was a postdoc, that created all kinds of interest, right? What am I going to do with a Z polarization? Now, that field only exists as Z polarization in the near field, at the place where you generate it, right? You could think of it almost like a dipole pointing at you, right? But what happens is that the intensity distribution associated with that field winds up having a narrow point spread function. So as a result, it's a handy tool for microscopy. The complement of that is the so-called azimuthal beam. And it creates a null when focused tightly in the electric field, but it creates a strong magnetic Z component on axis. No one's been able to, outside of theoretical work, no one's been able to figure out what to do with that, in part because you need matter that has strong responses in the magnetic component for this to respond at the frequencies of the optical field, okay? So this led to a lot of interest in work, but this is an example of a classically entangled field, right? And again, air quotes, right? Classically entangled field, whereby this mode, the spatial mode, is married to this polarization state. This spatial mode is married to this polarization. So we can't write this as a tensor product of the two. It's written as a superposition state. I never thought about that. You know, like, why would I think about that, right? And to some physicists, I know this is blasphemous, right? So I'd say take that tongue in cheek, because we know this is not truly quantum entanglement in any way, shape, or form. But the math here that we tried to borrow is what gives us some interest and insight. So there's work that's been done. In this particular case, this came out of Europe, where researchers were using the fact that you have this polarization diversity in the field to sense objects moving across the wake of the fields with high fidelity. You have work that was been out of my lab and one of our physics students, Rutendo, whereby we tried to exploit the polarization diversity to look at sensing. Sensing in terms of the physiological state, the amount of oxygen concentration that is in the body, ideally without bias to that. So there's been some interest in applications. So then let's take a look at another area whereby now there's something called space-time optics, where in this particular case what you have is an optical field pulsed, and you make deliberate, very specific correlations between the spatial frequency of the field. Remember, spatial frequency tells you direction, and the temporal frequency of the field. So you can think of it as wavelength. So each wavelength now is ascribed a particular phase function and direction. Right, so the gradient is going to give you the spread. You know, each wavelength goes in a particular direction. The SLM has a very specific hologram on there, and you synthesize a light sheet, something called a space-time light sheet that has very interesting properties. Namely, and many of you have probably heard of things like Bessel beams and Airy beams. This is another class of beams that is propagation invariant, right? So meaning that, you know, normally if I took this laser pointer and I shined it really far, it's going to spread because of diffraction, right? So so you can imagine here in the propagation invariant field, along the spatial degree of freedom, there's no spread. Secondly, in terms of the temporal degree of freedom, we have a pulse, right? So this pulse itself would not spread with the propagation through a medium. So we have something here that we thought was interesting, and we were curious over the years of sort of combining this so we can say, okay, do we have this interesting entanglement idea between space and spatial frequency and temporal frequency? Can we combine this also with the vector field, so you can actually have a space-time vector light sheet, right? So again, set up to create the pulses and spread the beam. We introduce a component here called a vortex wave plate, which basically, for all intents and purposes, all it does is it imprints whatever vector field distribution we care about onto the system. And we see here that we can generate space-time light sheets that have essentially the equivalent of the radial beam and the azimuthal beam. These are different polarization projections in terms of what you get from that. And we see that just like with the regular space-time field, the space-time vector counterparts, as you propagate, and again, this is a light sheet. This is the vector equivalent. This is so-called scalar equivalent, and we're propagating over some distance, over 200 centimeters. And we see that the spread within the beam is slow, and more importantly, is consistent with what you'll get with or without the vector piece imprinted on top of that. So it's great. And one other thing that's interesting about these fields is that they self-heal. So if people talk about Bessel beams are idealizations. Area beams, but you can generate them over short distances. Area beams take parabolic trajectories over some distances, and you place an object in their path, and what happens is that the field sees that object, and if you get just enough of it over the object, it reconstitutes, albeit with less energy, or less intensity, if you will, but it reconstitutes. That's what's referred to as self-healing. And we see the self-healing effect here. In this case, I think we had a fission line as the obstacle that we can also still get, and we still get the vector behavior here. So that's one dimension of the work where we were exploiting this entanglement. This one is even weirder, and I'll say just a few words very quickly about this. So this here was the idea of what we were referring to as coherence conversion. So if you think back a few hundred years ago, people were doing interference experiments. Okay, that's long before the invention of the laser. How did they do it, right? So they can take a candle light, they take natural light, put a pinhole, for example, and force spatial coherence, right? But your throughput goes down. If you have colored light, like filtered sunlight, like regular sunlight, then what you do is you put a spectral filter, and you get temporal coherence, if you will. But it's lossy. So we were curious about this idea of being able to do something a little bit different, whereby maybe we can take an unpolarized field that has high polarization entropy, but it's spatially coherent, take it through some type of unitary converter, right? So meaning that now if it's a unitary converter, it should be reversible. To be able to take it back and create a polarized field and make it incoherent, okay? And so here are the pieces to what we were looking at. And this started off when I was in Illinois, and then we continued this when I got here. So part of the trick to this work is that since we're using polarization, polarized field. So the polarization we'll say has 1 bit of entropy. So we need to make sure that the spatial degree of freedom is on par. So we take 2 points in the spatial field as opposed to the continuum of the field, okay? And what we have here is an unpolarized spatially coherent field. And lo and behold, what's fascinating about this, we have a polarization beam splitter, right? So a PBS is just a regular optical component, right? And what we do, when you take this unpolarized field, you split the arms. Vertical polarization is going to go along one direction. Horizontal polarization is going to go along the other direction. But look, they're polarized now, right? But what about the mutual coherence between V and H? So before, when we talk about unpolarized fields, what you have basically are, you can think of V and H having a random phase between them, okay? And so in this particular case, using this trick, this random phase now that was in the polarization now gets distributed between the 2 arms. So the output now is mutually spatially incoherent. And we argue that this has sort of this entangling operation, where if we looked at those simple 2x2 coherency matrices for polarization and space, we have, you can't write it as a product, this simple product idea. This is here unpolarized field, and here is a spatially coherent field. And this half-wave plate now, and you see, again, we have distinguishability. Vertical is married to this particular spatial mode. just the up direction in this picture, and horizontal to the other one. And when we introduce now this half-wave plate, which rotates the polarization, so now that distinguishability is gone, we say that's a disentangling operation, in which case we can write in a separable way. Now, why do we care about that? Well, here's an interesting thing you can do, right? So if you imagine, if you will, you have a polarization scrambler, right? Something that scrambles the polarization degree of freedom. You input light that's polarized, but spatially incoherent into the system. Can we transfer, for the purposes of transmission through that scrambler, can we transfer the randomness in the spatial degree of freedom into the polarization degree of freedom? Why we would want to do that? Because if the polarization degree of freedom is already random, and you're sending it through a randomizer, right, the scrambler, then it would be immune to it, right? And this is what we did here using this trick. And then we took a beam splitter at the outputs, and we were able to reconstitute high visibility fringes from, think of it as like the traditional double slit experiment that one would do. And I had a student who's now a postdoc in my group, Mitchell, show that you can do this in a reversible fashion along the spatial degree of freedom using the same idea. I'm not going to go through the entire detail here, but the way you want to think about it, in this case, our SLM, our spatial light modulator, has pixels that randomizes the wavefront. We send at the input light that is unpolarized, but spatially coherent. We get at the output light that's unpolarized but spatially coherent. This is why we can get the fringes. But the conversion is inside, and it's done without any filters, right? So that means we're doing this simple trick of combinations of the right combinations of beam splitters to move the entropy to the degree of freedom, essentially, that we don't care about, to deal with the obstacle in the way, and then recover it back at the output. So we have a manuscript where we're trying to exploit this for our communication protocol, because it gets more complicated, but it's super interesting, actually, what you can do with this. All right, now, final part of the talk. So here, many of you, especially in physics, are quite familiar with optical tweezers, right? And so the big deal with optical tweezers is that, yes, you can use light to move objects around if they're of the right size, right? And if it's the right index contrast. So in other words, if it's in the right medium. Okay, there are tricks that have been played over the years, if it's metal and all these other things where you can do interesting tricks of the beam. So Arthur Ashkin eventually got a Nobel Prize for this. It's been a very useful tool to biophysicists in particular. And additional tricks have been done where orbital angular momentum, not just, let's say, arrest in Brownian motion, but being able to take a particle and transfer optical orbital angular momentum so that it can spin around an optical axis or spin around its own axis if it's spin angular momentum. And so what trapping basically comes down to is the following. You have a field that is effectively, intensity distribution that's effectively inhomogeneous, right? So in the traditional case, you have, let's say, a focused optical field, and it creates a gradient in the optical intensity. And so what you have is a gradient in the intensity that translates into forces, reactive forces. So the light field pushes the object and exerts momentum. The object pushes back. And so you have stabilizing forces against the object being pushed along the axis, and that happens in 3 spatial dimensions. And it's super cool. It's gotten to the point now where undergraduate labs, you have a simple kit, you can build this setup, and it's very nice to see. The challenge with this is that the limitations are you typically have to have relatively high power, usually few milliwatts to a watt to trap simple, even dielectric spheres. And you're always going to be limited by the diffraction limit, how tightly you can focus. So when I was working a lot in plasmonics, we decided to explore this idea because it turns out, one of the big things with plasmonics is that it's all about localization, right? You can localize the field over very, very small areas. And so I was in engineering, and no chemical synthesis. For us, it's all about E-beam lithography. So we make these nice 2D structures of gold bow ties on some dielectric, basic glass. I mean, there's ITO there, and that ITO actually turns out to be very interesting, but that's another conversation. And you see roughly the dimensions here. So we got more than we bargained for when we worked on this. One, yes, in the hotspot region, you have very high field intensities. And those field intensities get even higher when you put this in an array structure based on the array spacing relative to the wavelength of light that you're coming in with. So there's coherent interactions that's happening. Now, that's one thing. Secondly, the problem with plasmonics we were trying to make into an advantage, which is that you get a lot of Joule heating, right? These things get pretty hot, right? Now, on ITO, there's a dissipation effect, but it's still pretty hot. Now, when you're doing trapping, you have, in our case, experimentally, some gasket on top of our bow tie structures. These are SEMs of our bow tie structures, thousands of bow ties, and the solution. So that fluid now gets hot. So you actually have convection happening. So it's a very interesting multi-physics problem. You have convection, you have, of course, local heating, and you have electromagnetic fields. So if you play it right particle sizes, right conditions, you can have a very interesting set of effects. So one interesting set of effects that we saw along the way was we can exploit this heating to do things like near-field writing. This near-field writing trick is accentuated when you place these antennas rather than just on a regular substrate. You basically place them on pillars. Like these are 500 nanometer tall pillars. So heat now, you don't have as great a heat sink as what we have in this particular case, and these things can actually get pretty hot, right? So the gold can start to reflow when you are excited with the light field, and that can show up as a plasmonic or a color shift. And this allows us to write in the near field. And if this was working, so we can do things like make movies, we call it Nano movie. By this time I was on sabbatical. This is what I was doing on sabbatical. Or make music. So if this was plugged in properly, you would actually hear a lullaby playing on this, all from this writing. Very simple ideas. So here, I'll skip over the whole thing about their plasmonic properties in terms of nonlinear optical properties. But here's what you see here, is that you have your antennas and you have the laser beam coming from the bottom side and it's moving the particles on the surface. And you can do that in a swarm. And one of the big things that's always been there about plasmonics is that these antennas can mediate light-matter interaction, right? So here you actually have a situation where you have some fluorescent particle that's off the antenna. When we drag it onto the antenna, you see the particle's fluorescence become stronger, right? So these are all effects that we can observe under the microscope, which is why we thought it was very, very cool. Now that's great. The problem with plasmonics for us was that it was a bit of a pain to make these different samples after a time. And we wanted to see if we can get some of the benefits without necessarily going through all the things we did with the plasmonic field. So I had another student, Chris Shange, who has been exploring using femtosecond lasers. So all the stuff we did before with the trap-in, we did a lot of that with CW, although we did a little bit combined plasmonics with femtosecond sources. So here we said, well, why not just trap directly with the femtosecond laser, right? Now, people have done that, and they've done that to the point where they think they know everything about trapping. Turns out they don't know everything because the parameter space for optical trap is extremely broad. Not high energy physics broad, but it's broad. And so you have a dependence on particle size, input optical power. There are other parameters that have to be taken into account. Right? So, but what's cool here, it turns out that there's some relative advantages that mirror what we can do with plasmonics. With this system, these pulses, 120 femtoseconds, 80 megahertz, that's the laser system we use for all the nonlinear optical imaging. Okay, that's the important piece here. And particles of the right size in the so-called Lorentz-Mean regime, we can now actually trap on the order of microwatts, tens of microwatts. And for reference, just to remind everyone, this laser pointer has a power of roughly 3 to 5 milliwatts. So microwatts is now pretty small, right? And this is what we were actually doing when we were doing all that plasmonic tweezer stuff. So we said, okay, let's see what we can do with it. If you're curious, this says something about the introduction of a transient force component associated with using energetic pulses. There's a sweet spot in terms of the rep rate and pulse width, in which this seems to work fairly well. This gives you some idea of the typical intensities that have been used in optical trapping. This was used in plasmonic tweezers, and this is where we are now with the femtosecond platform that we are doing. And so Chris showed quite well that she can trap bacteria that are moving around in solution. And what's fascinating about this, and you actually get a stiffer trap, what's fascinating about this is that you can look at something like the motility of the bacteria using this type of system, where, and this was done in collaboration with Anita Shukla's lab, my colleague here in biomedical engineering, where here you have sort of the control. This is the bacteria jiggling around within this solution, and here you'll see, I think the control in this case is gray, so this is just mapping the positions as a function of time. And then you actually turn off the trap and you watch what happens. You do the same thing with a continuous wave laser, sort of before and after trapping, right? And this is what you get. And then eventually with our femtosecond technique, we see that the motility mirrors closer to what the actual control situation is. So we think we are altering the biology somehow when we use a CW laser, even though they're on the same threshold for power, more or less, they compare it to using the traditional pulse laser. All right, now, I mentioned that this was all done on a microscope pulse laser. Well, instead of reading information, I said you could write information. So take everything I said before about reading second harmonic images and think about now just having a very simple system where you have some photoreactive material, like what you would do with photoresist. Now, it's a 2-photon effect, right? So you're dealing with actually a femtoliter volume. So wherever the photons are coincident in space and time, you're going to get cross-linking events. And you write that in 3 dimensions, right? So this is nanoprint. This is commercial now, right? This is what Nanoscribe sells, right? And this is what my student Jillian has been working on. And here you see we can take our platform, write our logo. We can mirror. So this is a second harmonic image of our tissues, and we can mirror that image digitally and print it out and look at this in bright field. And for us, we were curious about coming up with an algorithm to use bright field writing for in situ inspection as we write at any layer. And Jillian came up with a very clever algorithm that's used in additive manufacturing to do that, so that per layer, we can actually see as we're writing, because unlike a biological sample, we're controlling the information. We're placing it down onto the system. And then if you can do that, you can do image analysis. So what we realize is that we can take these 2 platforms and combine them, right? And that's what we spend a lot of time doing in my lab, is coming up with acronyms, right? So this is selective tweezing and immobilization of colloids, or STIC. And what we have here is this ability to now take whatever we want to tweeze, put it in a photoreactive material, place it, and immobilize the structures in that position, right? And here we decided to try this with few-layer graphene. This was in collaboration with my colleague Bob Hurt. Here, trust me, now after today, I'm also going to be harassing a few of you in condensed matter for your samples. But basically, this is few-layer graphene. We're just showing that we can tweeze and move it around. What's fascinating about materials like MOS2 is that you're not supposed to be able to do this, right? Because if here, this is from somebody else's paper, we show here, this is basically looking at trapping efficiency as a function of position. The yellow is the efficiency along the Z direction, right? So you should see a change in sign like you have in these cases with these other fields. So what we found an interesting trick to allow us to do this. So in this case here with graphene, we just show that we can take the graphene and photoresist. If this was water, 300 microwatts of power to move the graphene, okay? In photoresist, much more viscous. We have to put more power in, but we're using this femtosecond platform, and we can move the graphene around, and then we can place them, right? And what we're basically doing is we're anchoring it. We're polymerizing the region around the two, and this is few layer graphene. It's not really single layer graphene, but it's getting close to it. We can anchor it to the substrate, okay? And we do the same thing here, but what's interesting with the MOS2 is that it's really difficult to trap. So we stumble on something where we actually can take the photopolymer itself, create a handle, right? Create literally a handle, and the handle now you bring that, you optically tweeze that and bring it adjacent to the sample of interest, in this case the MOS2, and you can move it around. And so we show that we can do that, and we can place them and arrange them in any way that we care about, right? So this is a new platform that we have that brings together a lot of our interests. You know, the last piece you said, well, what about the multiphoton imaging? Well, yeah, you know, in this case, this is just beads that we do in 3D. We said, well, we know for MOS2, if it's odd layer stacking, it's non-central symmetric effectively, so it will give a second harmonic signal. And we saw that here where we have our sheet that we've polymerized. So we've had these MOS2 adhere to the sheet, turn the lights off, do the second harmonic probing, and we actually get the signal there. So we have a platform that would allow us to potentially move objects around, freeze them in place, and probe them. And I'll just say quickly, we can also do this with cells. Right, this is another dimension for the work. And I think I somehow got through all my slides. I will fast forward through this. I think you all get the idea. We have a lot of interest in the lab, and we love collaborating with the folks here. So I will stop here, and if there's time, take any questions.
Okay, so let's thank Kimani for a very inspiring talk. Do we have questions? Okay, Jay, of course, the most difficult.
Thank you. Very impressive range of work, Kimani.
Thank you.
Just one question about the pulsed laser trapping. It's amazing that you can trap with much lower intensity, sort of 2 or 3 order of magnitude. So, I mean, biological applications, it's wonderful because you have much less concern of damage to tissues and cells. So, but from the physics point of view, what is the sort of the reason behind it?
We still don't know. And the model that we put forth, even in our paper, is really based on the Rayleigh regime model. What makes us so interested is extremely controversial in the field. So long before I came to Brown, I thought there's no difference between trapping with a pulse laser generally or a CW laser because there are plenty of papers that say that. There are also papers that say, oh, there's some relative advantage due to this transient force component. It's an auxiliary force component that is there in addition to the gradient force. But we've yet to see a worked out model. And I spoke about some of this at Rochester sometime at the end of last year, and we're thinking of doing a collaboration with someone to try to work out what might be the reason behind this, because it's not clear to us either, other than the fact that, yes, you have this transient force component. If the rep rate is too high, it's the same as CW. Any advantages there are gone. Right, so you know there are games there, and it's the sweet spot in the size regime, which is on the order of the size of the cells, right? So this is why we think there's a whole new world. I skipped over a lot with optomechanics, right? Where you want to, if you want to measure with fairly good spatial resolution the mechanical properties of tissues or cells, often what happens is you're doing bulk measurements. If it's an AFM, it's a very noisy measurement, and there's a certain force regime that you have access to. But with this platform, you can directly, without using auxiliary, let's say beads, that you have to functionalize or anything, you can directly act on the cell, right? So it opens up more doors there that we think we will take advantage of, and that's the subject of Krishangi's thesis work.
Cool. Thank you.
Okay, other questions?
Dan.
Yeah, of course, Dan.
I'll ask like a seriously technical question if you don't
Sure,
mind.
no worries.
You're putting 50 milliwatts through a microscope. That's going to produce an intensity close to 10 to the 14, maybe 10 to the 15 watts per square centimeter.
Yep.
Why are you not ionizing? Why are you not field ionizing things at the focus? That's a huge intensity. I mean, how do you know that 2 photon is the only nonlinearity?
Yeah, no, it's a great question, a great point. And one thing I have to check, and I can probably tell you this for sure tomorrow, Dan, is whether that 50 is when I confirm that is 50 milliwatts into the microscope versus measured at the focus. But I don't know. That's a great point because you should get, your point is the ionization effects that would occur.
Just when you're at an intensity of 10 to the 14
Yeah.
there's an awful lot of nonlinearity.
But remember now, this is also in the photoresist itself, right?
Yeah.
But so, we wouldn't. So in water, if we went anywhere near that high, you start to see boiling, like we've seen that, right? But in the photoresist, I'm not sure as to what's going on. So the nonlinear effects, and by the way, the nonlinear effects in general is one of the things that people have brought up, I think, to Jay's question as to whether or not you have some type of interesting Kerr effect that's occurring that might explain why you actually have these auxiliary strengths. But that's a great question you asked, Dan. I'll see if I have an answer for you tomorrow. So that's the benefit. I'll have to come back and give you guys answers.
All right, maybe a last question. Okay.
I enjoy your talk. Thank you very
Thank
much.
you.
To answer my high energy colleagues' concern for burning something, the focusing point is diffraction limited.
Absolutely.
So for you have a UV laser, not UV, sorry, IR laser, right?
Yeah.
So we're talking about over a volume of a micron cubed space.
Yep.
So there is no energy density divergence or it is worried about. So the water is not boiling. So
That's a good point.
So that's one observation. The other one is concerning the answer for the pulse laser to trap. I think all you have to all there is just when you're during the pausing, when you're during the turning off period, the particle is not diffusing to anywhere. So as long as the particle is not diffusing out of focus, so you can cut off the beam as you want, and then you can still have the trapping. I think the sweet spot is the diffusion. So diffusion is related to diameter. So you can play the game there.
Yeah, no, that's a great point. And there's been some work where people have looked at this issue of sort of this diffusion time relative to, the pulses are coming roughly every 12 or so nanoseconds, you're basically sort of re-trapping it. It wants to drift away from the trap, and then it comes back.
Yeah.
Drift away,
So,
and so it comes back.
so you will see the difference if you try to trap, let's say, a 10 nanometer particle versus a particle of a half a micron. You can check.
That's a good
there.
point. I don't think we've gone as low as 10 nanometers. That's a great point. Excellent. And one of my colleagues at Rochester has a system where you can easily change the rep rate. So that's something that we actually want to play with and see what we get there. That's a great point, though.
Okay, let's thank Kimani Toussaint again.
Thank you. Thanks.